Thank you very much for the invitation. It is extremely honored to have this opportunity to do my presentation. I have nothing to declare. The first CAR-T trial in the United States was conducted in 2003, while first CAR-T trial in China was not until 2012. The United States and China continue to dominate the cell therapy development pipeline with a total of 791 and 695 agents in each region, respectively. The cumulative number of cell therapies in development increased by 31% in US and 40% in China in 2019, 40% and 69% increase in 2020 in United States and China, respectively. Let's take a look at the CAR T-cell clinical trial around the world. China is the country with the majority of CAR T-cell clinical trials. Up to March 2020, China had 460 clinical trials, US 286, UK 14, Germany 9, etc. This slide shows registered clinical trial for CAR T therapy each year and the publication of CAR T related study in China versus in United States. The red line represents USA and the green line represents China. You can see overall registered clinical trials are more in China. However, CAR T related publication, United States is way ahead. Planned sample sizes in each trial are typically larger in the United States than in China. In China, often started with investigator-initiated clinical trial and often are a single-centered trial. In the United States, 83% pipeline are developed in industry. In China, 60% in industry and 40% from academic center. Up to now, in hematologic malignancy, CD19, BCMA, and CD22 continue to be the most frequently targeted targets. Some new targets show a significant increase, such as GPRC5D for multiple myeloma, CLEC12A for AML, and CD7 for T-cell malignancies. Development of allogeneic cell therapy has increased sharply than autologous modality in the past year, with no major change in the geographic distribution in those modalities. A total of more than 50 CAR T-cell products from 28 domestic companies in China have been accepted by IND. Within those, a total of 39 have been approved for hematologic malignancy. So far, there's only 2 CAR T products approved for commercial use in China by CFDA. Both products are brought from United States. One is Axicell, the other is Rumcell, both for non-Hodgkin's lymphoma. You can see even though are quite expensive in China, however, compared to United States, still way less cost. Lu Daopei Hospital is a hematology hospital. Since 2015, we have been performing CAR T-related clinical trials. Just last year alone, we enrolled 394 patients into our different CAR T clinical trials. Our hospital is the largest transplantation center in China. Patients are referred all over from other cities. Just last year alone, we performed 1,168 cases. Within those, 70% are haplotransplantation. The other 10% or more from matched and related donor. Less than 10% from identical sibling. few cases of cord blood transplantation, less than 10 cases of autologous transplantation. Let's take a look at some of the CAR-T clinical trials that are performed in Ludao Hospital. Because of a time limitation, I will only focus on CD7 CAR-T therapy for treating T-cell malignancies. This paper recently published in Blood. A commentary also issued for our paper, CD7 CAR sword and shield. As you all know, effective treatment are still lacking for refractory relapse TALL, LBL patients. Once relapse, majority of patients often have slim chances of long-term survival. Compared with substantial efficacy of CAR-T therapy that have achieved in BALL patient, CAR-T therapy for T-cell malignancy is still being explored in the early stage of clinical trials. We developed the anti-CD7 CAR-T therapy with a novel platform. We conducted a phase 1 trial to investigate the safety and efficacy of CD7 CAR-T therapy in a refractory relapse TALL, LBL patient. For preclinical study, we did a comparison of 3 anti-CD7 CAR-Ts. One is NS7 CAR, is natural selection of bulk T cell transduced with CD7 CAR. KO7 CAR is transduction of CD7 CAR into bulk T cells ablated for CD7 by gene editing. NEK7 CAR is the CD7 CAR transduced from purified CD7 negative T cells. NS7 CAR had a significantly lower fold expansion than KO7 CAR and NEK7 CAR in vitro. NS7 CAR had a rapid and dramatic phenotypical transition from CD7 positive CAR to CD7 negative CAR T cells per flow cytometry study. CAR molecule expression in NS7 CAR was significantly higher than KO7 CAR or NEK7 CAR. Both NS7 CAR and KO7 CAR showed comparable cytotoxicity and release high level of gamma interferon and alpha TNF, but NEK7 CAR to a lesser extent. Both NS7 CAR and KO7 CAR conferred a robust protection against leukemia progression with a marked reduction in leukemia cell burden. While both are effective, however, the benefit of NS7 CAR is without needing for additional gene editing. With that, we applied NS7 CAR to our phase 1 clinical trial. So let's talk about more detail about NS7 CAR. As we discussed before, this is a natural selection of bulk T cell transduced with CD7 CAR. This is a murine-based CAR-T product with 4-1BB as a co-stimulated domain. Although the NS7 CAR did not show detectable surface expression of CD7 by flow after in vitro culture, significant CD7 mRNA and protein level were detected by RT-PCR and Western blot. as expected, no CD7 mRNA or protein expression was observed in NEK7 CAR or KO7 CARs. We confirmed that expression of CD7 mRNA from NS7 CAR persisted in patient on day 28 after CAR T infusion. So we think the CD7 molecule of many NS7 CARs are most likely antigenically masked or intracellularly sequestered by the CD7 CAR. So the possibility that a fraction of NS7 CAR T cell emerged from fratricide during the in vitro natural selection process could not be ruled out. From November 2020 to September 2021, we enrolled 20 patients into this clinical trial with a median follow-up time of 143 days. 3 patients received the lower dose, and 16 patient median dose, and 1 patient with a higher dose. The median transduction efficiency was 95%. We have 100% manufacture success rate, either CAR T derived from patient's own blood or manufactured from patient prior transplantation donor. The median age was 22 years old, from 3 to 47 years old. The median bone marrow blast was 21%, can be as high as 87%. And 3 patients had blasts in their peripheral blood. The median lines of prior therapy was 4.5. Some patients even up to 8 lines of prior therapy. 9 patients had extramedullary disease involvement, including 3 had CNS leukemia or orbital involvement, and 5 patients had localized EMD, and 4 patients have a diffuse extramedullary disease. And 5 patients were relapsed from previous transplantation. And 2 patients with ETP-ALL, and 3 patients had a still tall fusion gene. Characteristics of NS7 CAR products for each individual patient are listed in detail in this slides. NS7 CAR contain a significantly larger population of CD8 positive subsets. A total of 18 patients received autologous NS7 CAR T products. 14 out of those 18 patients, their malignant blasts did not express CD3. Therefore, their T cell of those 14 patients was sorted by CD3 magnetic beads. 4 patients were sorted by CD4 and CD8 magnetic beads. There were no detectable malignant cells in the final harvest NS7 CAR T products. In addition to flow cytometry study to confirm there's no malignant cells in the final products, we also performed the qPCR. Those are our representative cases using the qPCR, tracking some of patients who have a still tall fusion gene. Again, no detectable malignant cells. So use of either CD3 or CD4, CD8 selection coupled with in vitro natural selection and potent anti-leukemia activity from NS7 CAR could successfully exclude malignant cells from the final products. This is a busy slide represent our clinical outcome for the treatment. At day 28, we performed a complete evaluation for the bone marrow. You can see 19 out of 20 patients achieved MRD negative CR/CRi. Regarding extramedullary disease, 5 out of 9 patients with extramedullary disease achieved MRD negative status at a median time of 34 days, from 15 to all the way to 51 days by PET-CT scan. One NR patient, both in bone marrow and in extramedullary disease, did not achieve the CR. This patient also had still tau fusion gene and found to have a loss of CD7 on day 28. 10 CR patients subsequently underwent consolidative allo transplantation at the median time of 57.5 days. 7 out of those 10 patients remained MRD negative at the median follow-up time of 210 days up to 311 days. One patient died on day 82 from transplant-related mortality. This patient had a second allotransplantation. 2 patients withdrew on day 102 and day 147 respectively due to personal and financial issues. At the time of their withdrawal, they were still maintained in the CR. The other 6 CR patients without consolidation allotransplantation. 4 out of 6 patients remained CR at the median time of 54 days. However, 1 patient with follow-up up to 180 days were still in the CR. 2 patients relapsed early in the course. Both patient had a still tau fusion gene. This is one of the representative cases. You can see this patient who had T-cell lymphoblastic lymphoma with a diffuse extramedullary lesions. And this patient able to achieve complete remission on day 51 per PET-CT. These products have a good safety profile. You can see 19 out of 20 patients only had mild CRS, less or equal than grade 2. Only 1 patient had a grade 3 CRS. Median onset of CRS was 1 day and the duration was 13 days. Only 2 patients had a grade 1 neurotoxicity. Between the interval of a CAR T-cell infusion to the transplantation, no clear increase in infection rate. Only 1 patient had CMV reactivation. This patient had a prior allotransplantation. 7 out of 14 patients with allotransplantation subsequently developed GVHD. 5 patients had acute GVHD. 2 patients had both acute and chronic GVHD. The anti-CD7 CAR T-cell expansion and persistence were frequently checked both by qPCR and by flow. By qPCR, the median peak time was 19 days, ranging from 10 to 42 days, and the median persistent time were 90 days, all the way up to 195 days. By flow, the median peak time was 17 days, and maximum proliferation were 67.7% per flow. The absolute number of T, NK, and B cell of each patient were measured after NS7 CAR T therapy. CAR T cells and malignant cells were excluded from T cell counting. The absolute number of TB NK cells showed a temporary decline to different degrees for each patient during the peak of CAR T proliferation, then recovered to the baseline level with predominantly CD7 negative cells. However, decreased again shortly after allotransplantation. There were an adequate recovery of normal TNK after allotransplantation. The dynamic of CD7 positive T cells, NK cells, and CD7 CAR gene copy number in each patient are shown in these slides. On the left y-axis represent the percentage of CD7 positive cells and NK cells as determined by flow. The right y-axis represent CD7 CAR gene copies numbers by qPCR. Again, you can see during the peak of CAR T proliferation, the CD7 positive T cell, NK cell were diminished significantly. Conclusion. We developed this novel method to generate naturally selected anti-CD7 CAR T cells without gene editing or additional gene transfer. Our phase 1 study of NS7 CAR is safe and highly effective in patients with heavily pretreated refractory relapse TALL and LBL patients, including those with extramedullary involvements, those who had a prior allotransplantation, and those who had a high-risk subtype. However, patients with still tall tend to have a poor response and early relapse. Most patients are able to achieve MRD negative CR/CRi on day 28 and maintain progression-free. When relapse, loss of CD7 expression was observed. The majority of patients only experience mild CRS and no neurotoxicity. Certainly, more patient and longer observation time are needed for further evaluation, the efficacy and safety of the NS7 CAR. Well, CAR T cells are really amazing for treating some of those hematological malignancies. However, there's still room for improvement. Now there's an open clinical trial, even move beyond the conventional CAR T cells, have CAR NK cells, CAR macrophage, CAR from Treg cells, etc. CAR cells also moved beyond malignancies. There's reports for CAR T therapy for AIDS, HIV, for lupus, for autoimmune disease, organ transplantation, for heart failure, fibrosis, and even for aging cells. And those are listed CAR T clinical trial for treating non-malignancies in China. You can see also including AIDS, lupus, etc. So for the CAR T study and the clinical trial are truly teamwork, from preclinical study to clinical trials, every single steps. We are very proud of our team. I'm here with specifically thank you for Senlong Biotechnology Company. Thank you for Professor Li Jianchang. Thank you very much for your attention, and I'm open for questions.